Good morning, everyone. Continuing from the previous lecture, so we were discussing about some basics related to biophotonics, how light comes into picture, and biomedical tissues, etc., how they come into picture, and just introduction in terms of how biophotonics as a field is useful, or it's how it is being used for biomedical or techniques, biomedical devices, etc. So this is what we were talking about. Now just before we go to some basics of light, I thought we will also just get some idea about when we talk about photonics, photons or electromagnetic waves, they are going to interact with tissues and cells, etc., biological tissues, etc. So how this, now if we consider biological cells, or tissues, and maybe at a macro scale, organs, biological organs with which it will be interacting. In that case, we know that living cell is, any living cell is made up of approximately 70% water plus proteins and macromolecules, we can say, macromolecules. Now this macromolecules are of different types. One, as I told you, the proteins. One is proteins. Okay, then we can consider carbohydrates, then nucleic acid, and lipids. Okay, now if we consider proteins, basically they are nothing but amino acid, acids, and carbohydrates are concerning glucose, fructose, and nucleic acids are basically nucleotides, okay, and these are all polymers basically. Okay, so they are made up of chain of monomers, okay, polymers. On the other hand, lipids, they are not polymers, okay, and we will write are not polymers as such. When we talk about proteins, there are different types of proteins, okay, which are very important, and mainly one type is enzymes, and as we know, they take active part in catalyzing some of the metabolic activities, etc. Then there are structural proteins, okay. So for example, in hair we have keratin, okay, and if we consider skin, then we have collagen. So these are all proteins, okay. They are also, collagen fibers are also there in muscles. So not only skin, of course, it is skin, muscles, okay, and then even bones, tendons, okay, etc. These are all Then antibodies, basically these proteins, they bind to the foreign body, right, in assisting in eliminating those foreign bodies, etc. So these proteins are of special type, and there are proteins which are also known as transport proteins. Right, they basically transfer certain nutrients, certain, right, of material to tissues, cells. For example, let's say hemoglobin, right? So when we consider hemoglobin, they carry blood, right, oxygen through the blood cell, right, to different. So basically they carry oxygen, right, through the blood, through blood. Okay, so that's what is. Now if we consider next level of those tissues, so when we consider carbohydrates already, as I told you, see, carbohydrates are made up of, right, CHON, right, uh, right, CHON, atoms, right, in a interconnecting ring of CHON, etc., with a ratio of hydrogen twice that of the C and O. Okay, that is basically sugar, fructose, C6H12, O6, etc., starch, cellulose. So sugar, right, fructose, starch, cellulose are all, right, carbohydrates, and all nucleic acid, which is, right, uh, they store and process genetic and hereditary, right, information. So we know deoxyribonucleic, right, acid, right, deoxyribonucleic acid, right, then RNA, deoxyribonucleic acid, monomer of this, they all, right, come together. And there is one more important nucleic acid which is useful, which provides energy for metabolic activity, etc., is adenosine triphosphate. Okay, triphosphate, which is available in cells, some of the, right, active cells, for various, right, energy transfer, active metabolic activities basically. Now if we consider lipids, okay, if we consider lipids, they are non-polymeric, macromolecules but non-polymer type, such as fats. Okay, mainly fats, then phospholipids, phospholipids, okay, and steroids. Okay, they are basically insoluble in water, okay, and the fats provide long term energy, right, they they store and, right, provide long term energy and insulation to certain extent, right. And phospholipids, they are found in cell membrane, and used for cell to cell signaling. And steroids, right, steroids are basically cholesterol, right, then estrogen, right, etc. They also provide right, immediate some energy, etc., through this. So these are all various components associated with the cell as such. And cells are of biological cells are of mainly, right, of two types. One is prokaryotic, right, cell, 1 to size maybe 1 to 10 micrometer in diameter, okay, in diameter. And then eukaryotic cells, right, and they are 10 to 100 micrometer in diameter, okay. So basically bacterial cells, they come under this prokaryotic, right, cell structure, while eukaryotic cell structures are animal cells, plants, right, fungi, and a protist, right, which are one-celled structures, etc. So they all come under eukaryotic cells, and they all contain, as just now what we discussed, right, proteins, then lipids, carbohydrates, and nucleic acids, etc. Right, and tissues are made up of these cells, right. So tissues are nothing but ensemble, right, ensemble of cells to carry out specific functions, right, to carry out specific functions. Now the tissues are of again different types. So epithelial tissues, connective tissues, right, muscle tissue, and nervous cells, okay. Now when we consider epithelial tissues, they are basically they are surface, they, right, they come into picture outside surface of the skin or inner structure of the organs. So outside surface the of skin or inner surface of organ. So these tissues, they are right, they have certain functionality, surface of the organ, right, of organs. I can see maybe L, okay, of organs, okay. And what functionalities do they provide? So they protect against, right, mechanical injury, against mechanical injury or external bodies, right, mechanical injury, invading microorgans, external, right. So they protect against invading, protection against invading microorgans. Okay, and they also, some of these cells also, right, secrete mucus. They, right, mucous layer they provide for lubrication, moisturization, etc. Lubrication, moisturization, etc. Right. When we consider connective tissues are, on the other hand, they are binding, right, structures. They provide binding structure, right, or fibrous connectivity, connective right? So they are present in bone, cartilage, right, and even blood, right? So they also right, and they are important in blood tissue. On the other hand, muscle tissues, and they are long, right, and excitable cells. Okay, so excitable cells. So for example, cardiac tissue, and cardiac tissue, skeletal tissue, right? These are all skeletal muscle tissues, of course, muscle tissues, etc. Right? They provide, right? They are excitable for certain functionality, so that is taken care, right? And nervous tissue. Oh, I'm sorry. So they are present in central nervous tissue, that is brain and spinal cord, and peripheral nervous tissue, peripheral nervous tissue, that is cranial and spinal nerves. Okay, so they are useful in transmitting, right, certain signals for various right activities. So from cells we come to tissues, and then from tissues it's organs which come into picture, right? Now organs are of different types. Okay, around 70 different organs are present in human body, right, in other animals, etc. So they are of various size and shapes, and they have different functionality, as we know, right? Bladder or brain, right, eyes, right, heart, kidneys, liver, lungs, skin, spleen, etc., etc. Okay, and these are all collected, means they are made from different tissues with certain functionalities, correct? Certain functionalities. The functionalities include, let's say, tissues in stomach, right, digestive tissue, right, or cardiovascular tissue related to heart, vascular tissue, right, skeletal tissues, nervous tissues, nervous right activities, then respiratory activity, right? So they have certain organs will have certain functionalities, endocrine and lymphatic, right, and lymphatic system. So so all these organs, right, and cells at tissue levels, etc., are the ones which are responsible for various metabolic and physiological activities related to uh, right, human body, etc. And now we are going to focus on optical approaches, right, optical approaches in order to, right, understand sometimes, right, for sensing of this organs or tissues or cells for certain activities, or monitoring, okay, certain activities, or for diagnostic purpose, okay, diagnostic purpose of certain pathological conditions which may right disturb the functionality of the particular organ, then we may use optical approaches in such, right, cases, which is diagnostic, and therapeutic applications, right, therapeutic applications. This here therapeutic applications are basically related to interaction of light with the tissues, and then modifying certain characteristics of the tissues, etc. Okay, so these are all right aspects where optical approaches right are very useful. And uh, right overall, right, so if we consider optical approaches for, let's say, biosensing, right, and probe applications, then right, broadly, right, how are they dealing with? They may be dealing with tissue pathology, right, to understand characteristic of the tissue, malignant tissue, or right, tissues which are normal tissues, etc. We can characterize them. We can, right, use the optical technique in order to characterize this tissue through endoscopy as well, so that it can be in vivo endoscopy, or it can be even in vitro with tissue, right, pathology characterization, etc. We can use optical techniques to detect certain molecules, and not only detect, right, we can use this approaches to even measure concentrations and various characteristics of this. So molecular concentrations can be, right, estimated using this uh, molecular concentrations. We can measure molecular concentrations, right, then measure physical changes, right, measure physical changes, for example, right, body temperature or temperature, right, temperature, pressure, stress, strain, etc., right, then measure features of the cell, right, features of cells in terms of, let's say, proteins, right, DNA characterization, or other macromolecule, right, characterization. It is possible, right, by using this optical techniques. We can measure oxygen, right, it can come under certain other molecules, and oximetry or blood, right, characterization. Okay, and as I told you that these are the approaches which are related to sensing. However, is we can also make use of right, light for treating this, right, treating uh, right, cells, structures, or tumors. Okay, tumors, etc. So these are the broad approaches which are associated with optical techniques, right, concerning the biomedical applications, and uh, we will be right, studying various such approaches in next few right lectures, and understand how they can be used in order to build different biophotonics devices as such. Now before right, we go towards those uh, right, measurement approaches or probing approaches, we would also like to learn about some basics of light or electromagnetic wave, in general, I will not go into very much in detail, but in general, some of the specific aspects related to light which will be useful, that we will be studying in the next few lectures. We will start with how this electromagnetic wave is what is this electromagnetic wave in general, and then how it propagates and how we characterize this electromagnetic wave. Then we go a little further of how this light propagates through optical fibers, waveguide structures, right? What are evanescent waves? How this what are the different light sources which are useful for biomedical applications, including lasers? What are their characteristics, etc. So once we know those aspects, so when we are building any biophotonics device, etc., it will be very useful for us to select or decide about appropriate type of light source for different for certain applications, etc. So now this light or is basically it can be described with an electromagnetic wave. Okay, so electromagnetic wave. Now what is electromagnetic wave? It is basically, electromagnetic wave is made up of electrical field, electric field, and right E bar, sorry, once again I'll write down E bar and magnetic field. Okay. H bar. So this electromagnetic wave was first described with the help of Maxwell's equation. Maxwell's equations. Okay. And which are the Maxwell's equations which basically were used in order to describe this electromagnetic wave are the ones which are in, for example, if we consider this electrical field and magnetic field vectors in free space. Okay, so in free space, when we consider then first del cross E bar is minus mu 0 h by dt. Okay, so this is how electrical field spatially varies with respect to the magnetic field, temporal variable magnetic field. What is mu 0? Mu 0 is nothing but, we can say, magnetic permeability. Okay, of a free space, right? And next equation, Maxwell's equation, is del cross H bar. How magnetic field varies in space, and it in turn depends upon temporal variation of electrical field. Okay, so they are interrelated with each other, this two equations indicates that. And then so where epsilon 0, okay, is electric permittivity, okay, of free space, in this case free space. Okay, so and then del dot E bar is 0 in free space, and del dot H bar is also 0, being a monopole. Okay, monopole, so is not possible, it will always be in dipole characteristics it will be with. So this is the way that it is expressed. Okay, now this equations, okay, are important in the free space. And when we consider a medium which is linear, okay, homogeneous, isotropic, dielectric medium, right? So what we will observe that del cross E bar can be expressed by, right, where B bar is mu 0 mu r into H bar. Mu r is the relative magnetic, right, permeability, and del cross H bar is minus sorry is equal to d by del d bar by d del t. So this d bar is nothing but represents electric flux density. Okay, flux density, right? In absence of polarization, it is equal to epsilon 0 epsilon r e bar. Okay, e bar it is, and so d bar is epsilon e bar. Epsilon is nothing but epsilon 0 epsilon r. Okay, and B bar is mu H bar, where mu is nothing but mu 0 mu r. Okay, so that is the case. Now when we consider now what these equations they represent, they represent basically that how electrical field vector is associated with the corresponding temporally varying magnetic field vector, or vice versa, etc. And how will they help us in explaining, right, the electromagnetic wave? So basically, as per Maxwell, right, but how basically Maxwell equation was derived is, we know, right, that del cross E in free space from the previous definition is minus mu 0 dH by double H by double T, and del cross H bar is epsilon 0 double E by double T. Then using and right, other two equations, we can, right, derive the electromagnetic wave, and for that we use vector calculus approach. So we know that del cross E bar, okay, can be given by del dot del E bar minus del square E bar. Okay, now what is right, del cross E bar? So if I use, right, del cross E bar is equal to minus mu 0 double H by double T, and use it here, then del cross E bar can be given by minus mu 0 double del cross H bar, right, double T. So which is basically is equal to minus mu 0 epsilon 0 by double T square. Correct? If we substitute this thing. Now del del dot E bar in free space is 0. So what it implies, these two right equations, if we substitute in this right equation, let's say right, then what we will obtain if we substitute this equation and this equation and this equation, what we will obtain is del cross E bar, which is equal to minus del square is y, because del dot E right is 0. So del square E minus del square E, and this is given, and if we substitute del cross E as this value, then we get del square E bar is mu 0 epsilon 0 del square E y, right, del t square. Similarly, we can also derive del square h bar is mu 0 epsilon 0 h by double t. Okay, these two equations. So these two equations are it's correct, yes, there. These two equations are basically electromagnetic, represents electromagnetic wave with in a general form del square u bar is equal to, or if we bring this term on this side, minus 1 by c square del square u by double square u by dou t square, right, and URT means basically, I'm sorry, so this is the representation where C, here I'll write with C0 square, where C0 is nothing but 1 upon square root of mu 0 epsilon 0. Okay, so this is a general expression for an electromagnetic wave when it propagates through the free space. It is the spatial variation in this right field and the temporal variation, right, they are interrelated with this equation. Now what I told, it's basically when we consider linear, homogeneous, and right, when we consider linear, isotropic, and homogeneous medium, in that case, right, a small variation comes into picture based on the polarization, right, of this, because a dielectric medium, now in case of dielectric medium, the polarization field, okay, so in case of right a linear, what is the meaning of linear, right, I'll explain here. So basically linear field is where vector field, polarization vector field, okay, vector or polarization vector field is linearly, right, related to electrical field, okay, in space and time, electrical field, okay, this electromagnetic wave. So in case of a linear, homogeneous or non-dispersive, and non-dispersive, homogeneous, isotropic dielectric medium, polarization comes into picture and it is linearly related to this. And what is the meaning of non-dispersive? Non-dispersive means response is instantaneous. So when an electrical field interacts with the material, the response will be so instantaneous, and it will produce a polarization vector. This polarization vector in a medium, it can be expressed by epsilon 0, which is a permittivity constant, right? Then chi, linear component, right, is expressed in electrical field. It is linearly related to the electrical field, okay, polarization vector. And it can also be expressed by, and because this in turn will give you electrical field flux density, which is epsilon E bar plus P bar, or epsilon bar plus 0 chi E bar, right? So it can also be expressed by epsilon 0 1 plus E bar, right? So this will be the electrical field density, okay? So the electrical field of flux density, which is epsilon E bar, where epsilon is nothing but epsilon 0 1 plus, right, E bar, right, and B bar is mu H bar. So similarly, of course, similarly to that of the electrical field. So linear, right, dielectric medium means polarization vector is field proportional to, linearly proportional related to electrical field vector. Non-dispersive means response is instantaneous. Homogeneous stands for, right, material characteristic is, right, uniform. So relationship between P bar and E bar, right, is independent of position R, okay. Anywhere this relationship is independent. And isotropic means the characteristic of this material, right, in different directions is same, okay. So this is basically the medium exhibits same behavior in all the directions, exhibits same behavior, okay. um, so if the material is not isotropic, then this electric permittivity values, right, relative permittivity epsilon r may vary, right, depending upon the direction. We can say, we can think like this: medium exhibits same behavior irrespective of the direction, in all directions, right, for all directions, okay. So when we consider this relationship, right, in a dielectric medium, and again we can make use of, right, Maxwell's this equations based on this, and derive the relationship, right, between, means based relationship in the form of a Maxwell's equation, we will end up with the similar, right, outcome, where del square u is minus 1 by c square dou square u by dou t square, which is equal to 0, where here now c is nothing but 1 upon square root of mu to epsilon, okay. In the previous case it was 1 upon square root of mu 0 epsilon 0, while here c is just, what is mu we know, which is mu 0 mu r, relative permittivity, and epsilon is epsilon 0 epsilon r, okay. So if we right, for a right, if we consider this, right, then the relationship between C0, which is 1 upon square root of mu 0 epsilon 0, and C, which is 1 upon square root of mu epsilon, and this, right, relationship. So this 1 minute, and this gives C0 by C is equal to, right, epsilon by epsilon 0 mu by mu 0. Okay, and for a non-magnetic material, mu right is equal to mu 0, because mu r is 1. In that case, we can write for a non-magnetic material, C 0 by C can also be expressed by epsilon 0, which is basically square root of epsilon r. And you know what is epsilon r? It is nothing but square root of 1 plus this chi, right? Factors, etc. So now what this right form expresses, so basically this equation, right, is representation of Maxwell's equation, right, where you can be an electrical field or a magnetic field, and how it propagates, and right, the spatial relationship with temporal relationship is given. Now if we try to see this, so if we consider a plane parallel combination, it's just now I did it. So what we see that a magnetic field which is sinusoidally varying and right, propagating, will generate a corresponding electrical field, right, based on the Maxwell's equation. And this magnetic field, electric field, which is also varying sinusoidally, will in turn again generate a magnetic field, right, and this right field, and it's an instantaneous right process, and the this electromagnetic field, they will be, this electrical field and magnetic field will be orthogonal to each other, and the direction, right, of the energy propagation will be perpendicular to this two vectors. So it's a cross product of, right, E bar electrical field and magnetic field, a cross product, and in turn it will propagate, right, in the z direction. So let's say a magnetic field is oscillating in, right, y direction, y, right, electrical field is oscillating in orthogonal x direction, then the resultant, right, of wave propagation or the energy, right, propagation will be in the direction perpendicular to the cross product, basically perpendicular to this oscillating waves. Okay, which is so that is the way that, and what would be the speed, right, or the phase velocity, how this phase, right, changes with time. The phase velocity can be, right, given by this, right, form. So electrical field in terms of in time scale, right, is given by this, right, sinusoidal wave, where frequency is f is equal to 1 by this 1 cycle time. Okay, and the correspondingly in terms of spatially wavelength, right, also varies, where it electrical field in terms of its right propagation of this, right, electrical wave is with period equal to lambda in, right, which is nothing but the wavelength, and its propagation constant accordingly is 2 pi by lambda in free space. In the above this figure, top figure, frequency comes into picture, right, considering the temporal variation with time, and in the bottom figure, a wavelength comes into picture, considering the spatial variation of this electrical field amplitude with, right, distance z. So These aspects, right, based on standard our right equation, m e exponent of I omega t I phi, we can, right, we can determine the phase velocity, which is basically um, um, indirectly, right, which gives us the phase, the speed, right, with which the phase, right, propagates, and that can be estimated by this fact, right. So for change in phase with respect to time, in case of, right, when we consider the temporal behavior, it is omega, and change in phase with respect to distance, when we consider in wavelength term, it is k, right, propagation constant. And this omega divided by k will give us the phase with which it is varying, right, and v phase. And if we right, see these two equations, basically, um, right, it is this v phase is associated with the 1 upon square root of mu into epsilon, which is nothing but the speed of electromagnetic wave, right, in dielectric, either in free space or in dielectric medium. Accordingly, the value of mu and epsilon will, right, change. So the spatial and temporal, right, relationship is important. Now what this right, the electromagnetic wave which is propagating, and basically the energy which is the, right, being transferred along the perpendicular direction, so is basically is given by the flow of electromagnetic power, is governed by the Poynting vector E cross H bar, right. Electrical field, let's say it's oscillating in sorry, magnetic field, let's say it's oscillating in y direction, electrical field simultaneously oscillating orthogonally in the x direction. And this right, sinusoidal, this oscillation, right, results in right, propagation of this electromagnetic wave perpendicular to this and given by E cross H bar, right, in this. So S is also known as a Poynting vector. Hence the intensity of the light, right, can be given by the magnitude of E cross H bar. So it is half a time average, so half of real part, because being a sinusoidal, right, generation wave, intensity will also be the varying sinusoidally while propagating, and that is half of right, real part of E cross right, H bar. And there are two terms associated with this, right. One is energy density term, okay, energy density term, and the other one is the intensity term. Now energy density is basically given by the averaged energy density is given by right, energy carried by the electrical field and energy carried by the magnetic field, right, which is oscillating magnetic field. They add up to provide us the average energy density of this electromagnetic wave. So mathematically it can be expressed by either of these two terms, being E and H, amplitude wise they are identical, right, in nature, and hence they can also be expressed by this, right, term, where E square can also be expressed by E into E conjugate of, right, electromagnetic wave, electrical field conjugate, and hence with little, right, simple further simplification, we will end up with average energy density of the electromagnetic wave, which is given by this term. On the other hand, intensity which is E cross H bar, and if we derive based on the previous condition, if we derive intensity will be given by this term. So the relationship between the average energy density of the electromagnetic wave and the intensity of the light is given by I is equal to c into rho, where c is the speed of light either in free space or in medium, depending upon through which the electromagnetic wave is propagating. And so the time average power density flow results from the transport of the time average energy density, basically. Intensity results from energy density with the relationship I is equal to c into rho. Okay, so it's a very important term. And one more aspects associated with this, when we consider electromagnetic wave, we can also express it in terms of photons. So in terms of photons, that means the photons which are propagating through the medium, let's say photon energy is h nu, and number of photons per cubic centimeter are n, which are propagating through the medium. So this energy density rho of the electromagnetic wave can also be represented by nh into nu, where n is the number of photons per cubic centimeter, h nu is the photon energy. So this nh into nu and multiplied with c is the speed of light will give us the power density of this electromagnetic wave, or corresponding to the photon particular photon frequency. What is the power density term? It can be derived, basically we can express. And another aspects associated with the previous, see, as we understood that speed of light in a free space is c0, which is 1 upon square root of mu 0 epsilon 0, while speed of light in a dielectric medium is given by 1 upon mu epsilon. And this ratio of this is also expressed in terms of, so ratio of c0 divided by c is also expressed by the term refractive index of the medium. Okay, so refractive index of the medium is of material is can be determined based on the speed of light in free space and the speed of light through the same medium, through that medium will give us the term refractive index. And this refractive index corresponds to it is, of course, it depends upon the material characteristics of this medium. So different medium, they have different refractive indices depending upon the material characteristics and constituents. So so air, it is 1, while water it is 1.33, and if we consider, let's say, tooth enamel, it is 1.62. If we are considering epithermal tissues or epidermis, then it is around 1.34, etc. Now this refractive index plays an important role as far as material scattering is concerned, dispersion is concerned, when the propagation of light is through the medium. Correct. Now this electromagnetic wave, when it in general is expressed by electrical field vector and H vector. Both are oscillating simultaneously, but in different orthogonal planes that we understood. Now this most of the light what we see in surrounding is not unpolarized. Now what is the meaning of this polarization? It means when see light, this polarization aspect associated with the electromagnetic wave is based on the the plane of oscillation of either electrical field or a magnetic field. Okay, so if we can say that an electromagnetic wave is linearly polarized if there is only one, right, electrical field, a plane of electrical or electric field, and correspondingly orthogonal plane for magnetic field, right. So it is represented with the plane of oscillation of either electrical field or magnetic field. Maybe we can consider, let's say, the plane of right, oscillation of electrical field as the reference. Okay, so linearly polarized light can be represented with the right plane of oscillation of electrical field, and it will have only one electrical field, while unpolarized light will have multiple such, right, electrical field vectors which are oscillating in right, in different angles, with different angles, right. That means it will have, so electrical field will be of unpolarized light will be oscillating in uh, right, different planes. Okay, so all different planes. So one electrical field oscillating in this plane, another one in this plane, right. So this electromagnetic waves consisting of many such electrical fields oscillating in different planes. Okay, so what happens, the light is treated as unpolarized because at any point of time, right, we will see that different electrical fields in different directions, right, are are oscillating in this, right, in this particular plane. And now, of course, correspondingly the magnetic field can be represented. So this unpolarized light is the one which we see in nature in general. However, linearly polarized light has one well defined, right, electrical field oscillation plane angle, and uh, that particular angle, right, determines along which this, right, linear electrical field is oscillating, and correspondingly at 90 degree the magnetic field, right, would be oscillating, right. So magnetic field at 90 degree would be oscillating. So and the right, and that is known as linearly polarized light. So it will have depending upon, right, how we consider when let's say, for simplicity, let's say this is the plane of incidence of electromagnetic wave, and if the electrical field of this electromagnetic wave which is propagating in this right plane of incidence with electrical field perpendicular to the plane of incidence, right, or parallel to the plane of incidence, right, will determine right, in how this electrical. So if there are two electrical fields which are orthogonal to each other, such, and they are both are electromagnetic waves linearly polarized, right, electromagnetic waves, one electrical field oscillating perpendicular to the plane of incidence, the other one which is oscillating parallel, let's say, to the plane of incidence, then it both are linearly polarized line, and the resultant, right, electrical field vector which would be, right, oscillating will be resultant of this Ex and Ey vector, and right, accordingly the resultant electrical field will be oscillating in a linear plane in a one particular, right, direction. Now when we, so depending upon, if there is only electrical field oscillating only in one field, only one component if we consider, then the linearly polarized light is represented by this form, right. It has a 0 y component, while it has only x, right, along the x direction, the electrical field component. And if there are two components, both are orthogonal to each other, electrical field vectors, two electromagnetic waves, right, with linearly polarized electromagnetic waves, then the resultant, right, vector field, which is given by this E bar xyzt, is given by the term along x bar direction and another along y bar direction. This is the resultant two components. So the resultant field would be oscillating in a plane, right, linearly polarized in a particular plane, right, with the theta as the angle with respect to the x-axis and 90 minus theta is the y-axis, right. So that is the way. Now slight variation. Let's say we have two electromagnetic waves with one of the electrical field oscillating, that means with two electrical fields oscillating in oscillating perpendicular to each other, but with some phase difference in terms of its right phase, although the phase velocity is the same, that means the wavelength is the same, but these two electrical field vectors are perpendicular to each other, and at the same time there is a phase difference of pi by 2 or pi by 2 plus 2 m pi, where m is the integer. So with this phase difference, when these two waves are propagating, we can see that the resultant electrical field, right, resultant field due to this two electrical vibrating electrical oscillating electrical fields, orthogonal electrical fields with a phase difference of pi by 2, will result in uh, resultant electrical field varying, right, such a way that as these two electromagnetic waves propagate through the medium, the electromagnetic wave will propagate like a circularly polarized wave. Okay, so both Ex and Ey are equal. Okay, magnitude wise they are equal. Ex and Ey components are equal. The difference is the phase between them is right 90 degree to each other. Okay, the phase difference, relative phase difference is 90 degree, and hence the resultant electromagnetic wave which is generated will have electrical field, resultant electrical field varying like a circularly polarized, right, it is circularly polarized line, and depending upon their right direction of this electrical field oscillations, they will have either right circularly polarized line or the left. Okay, so right means it the electrical right field intensity, right, will it will propagate along this direction. Electrical pointing vector will be the resultant of this vibration, orthogonal to this E waves, E vectors. However, the electrical field, right, amplitude would be varying in this way, and right towards in an anti-clockwise direction. So it is right circular or clockwise direction, so left circular, circularly polarized wave. Now in case if the electrical field amplitudes are not equal, or if there is a phase difference between Ex and Ey other than this pi by 2, right, or a pi, if that's the phase difference, what we will see as basically instead of a circular propagation, when there is a phase difference delta other than the pi by 2, we will see that the um, electrical field, resultant electrical field due to Ex and Ey, whose magnitudes may be different, as well as the phase relative phase difference is not pi by 2 or some other angle other than 0 degree and 90 degree pi by 2, we will end up with a electrical field vector rotating right, of this electromagnetic waves will be resultant electrical field vector would be forming an elliptical, right, progression when it is energy is propagating orthogonal to that. While it is getting propagated, the electrical field would be propagating in, right, that direction. So these aspects are important in order to, when electromagnetic wave interacts with the tissue material, etc., depending upon the polarization state of this electrical field, right, we may observe sometimes a slightly different phenomena, and that may be related to birefringence of the material, etc. So what it is, also just with a very short simple way, if I can explain. First of all, as I told you that let's say linearly polarized light oscillating in x direction, it is we know in particular x, right, direction, the electrical field, right, amplitude is varying, and linearly polarized light at an angle theta, it will have this. So theta with respect to x, and then, right, then that indirectly it has it can be represented by two electrical field vector, one along x direction, one along y direction. Resultant is oscillating in, right, in theta direction, and um, right, if they are in the same 90-degree phase difference with the same magnitude, these two components, then depending upon the direction of this electrical field, right, vector, we will get right circular polarized line or left circular polarized light. Okay, and this depending upon the right material characteristics also through which this light is propagating, we may be able to observe the change in this polarization state as well. And that depends upon the optical this material, right, or the system. If optical material is such that it is having uniform, it's homogeneous and isotropic, that means refractive index in any of the direction is going to be the same, then electromagnetic wave, when it propagates through this optical system, if it is a circularly polarized light, it will remain circularly polarized light, right, because the refractive index observed by the electrical field oscillating in the y plane and the electrical field oscillating in the x plane, both will observe the same refractive index. That means the same speed with which both these waves would be propagating, and the resultant circularly polarized light will remain circularly polarized light, or linearly polarized light at an angle theta will remain linearly polarized light as it is. However, if the material is not isotropic and it has different refractive index depending upon the right electrical field oscillation, this direction, depending upon that direction, then vertical, let's say, component of electrical field may experience different refractive index than the orthogonal horizontal component, and hence when the light is electromagnetic wave is propagating through this medium, there will be a phase change, a phase shift, right, because the speed with which the electromagnetic wave corresponding to the vertical oscillation of the electrical field and will be different from the horizontal, right, oscillations. So both linearly polarized light having different refractive experiencing different refractive index would propagate with different velocity and phase velocity, and hence there will be a phase difference, and that in turn will result into an elliptical right, basically polarization state. So these aspects are also important associated with the type of material. If the material is birefringent material having different refractive index in different directions, we will observe such right birefringence characteristics using the electromagnetic waves oscillating in two different planes orthogonal to each other. The resultant will see that electromagnetic waves with a phase difference depending upon the birefringence characteristic of the material, we will be able to observe such phase difference. We will also use such right aspects characteristics for characterizing certain tissue characteristics, etc. Whenever we come across, I will try to once again explain you how exactly these terms are right are useful. um, Now, I will not go into this details, but as we can express this propagation of this polarized light using Jones matrix, Mueller matrix, and understand how this linearly polarized light propagates. And if it is linearly polarized light with two electrical field vectors orthogonal to each other but with different phase, how the resultant polarization wave will emerge, that also we can estimate using means mathematically with different forms based on their angle of this rotation of this electrical field vector, we will be able to determine. I am not going into this details, you will be able to study these aspects even when we come across such aspects. We will also try to then at the time try to see how exactly we can use it. In the next class we will try to learn how this light which is incident on biological tissue or cell, and how what we observe in terms of reflected light, in terms of transmitted light, in terms of scattering, absorption, etc. Those aspects we will try to see, particularly concerning the basics of the light, how the reflection and transmission comes into picture is what we will try to study in the next class. Thank you very much.